the only person in this room whose book I own. I don't buy a lot of books, so you should take that as a compliment. I looked up his website and I was fascinated by his productivity because I went through a whole page and we got back to 2016. So he's been extremely active. And I notice he's gone from the social insects to trying to understand Drosophila. And not only that, we are going to hear about a very, very controversial topic, which is emotions in insects. So Reuven, come and tell us all about it.
Thank you, Jennifer, and thank you, of course, Stephen, for organizing this wonderful program, and thank you all for coming. When I was invited to talk, I decided that instead of talking about my lab's research in the past few years, I would like to talk about something I would like to do more research about, and I like other people to do more research about because it's important, which is insect emotions. Before I switch to talking about it, I want to talk about pain in infants. You'll see in a moment why. Remarkably, until 1987, the medical convention was that infants do not feel pain. In 1987, a landmark paper said, of course infants feel pain. How could you think otherwise? So this means that infants that had surgeries before 1997 likely did not get any anesthesia or pain relief after the surgery. The result of this landmark paper from 1987 was that medical organizations definitely accepted the fact that infants do feel pain. But the medical profession changed very slowly. For example, here in Canada, a survey done in 1990 found that only 4% of physicians used anesthesia when they performed circumcisions. The reasons were variable. I would say the vast majority did acknowledge that the infants feel pain, but it was more a matter of being trained in providing anesthesia, fear of negative aspects of anesthesia, but that's a fact. So what's going on now? Just for information, circumcision, as you might imagine, is painful. There are data about it. There are long-lasting effects of the pain from circumcision. The prevalence is 40% of male infants that are born get this operation, but still the majority of them undergo the operation without anesthesia. Why? We have a tendency to first underestimate individuals that cannot talk, that cannot tell us this is painful. And second, even when we appreciate that they do feel pain, we still do whatever is convenient for us because they don't have the power to tell us it's painful, don't dare do it. So we have some issues, which hopefully we can talk about later today. Okay, so this is just quite instrumental example about pain in infants, and I'm not going to talk any further about it. In the past 20 years or so, I've taken an approach I like to call cognitive ecology. It's summarized in 2 edited volumes, and also in a very recent special issue of Animal Behavior titled Cognitive Ecology. It's online. It will be published formally later this year. So what's cognitive ecology? I like to define the cognitive system as the structures and processes concerned with the acquisition and use of information. Ecology is the study of interaction between animals and their surrounding biotic and abiotic environment. Such interactions can drive evolution, defined as a change in proportion of individuals differing genetically in a variety of traits over generations. Evolution is not a theory, neither abstract. You can go to the lab, come to my lab and you can see evolution in action within literally a couple months. We see an example of flies selected to prefer light or avoid light, and you can see that within a few generations, which would take a couple months, you see evolution in action. So evolution is very real, and you can see it over a short duration. Overall, the general approach is driven by the logic that integrating mechanistic information with evolutionary ecological insights can advance research on cognition in all animals, including humans. And finally, after this relatively long introduction, I would like to give an overview of my talk today. I'll give some definitions related to emotion and pain. Then I will talk for a while about the highly conserved mechanisms of pain, evolutionary biology of pain, and hopefully I will have time to switch to a more positive topic, which is pleasure. To guide their behavior, animals assign internal representations of value to stimuli and states. For example, the rat here on the left gets some positive internal representation from this wonderful cupcake. I know some other people who would have the similar rewarding internal representation. On the other hand, this generates a very strong negative internal representation. We refer to such internal representations as emotions, and we can use objective terms such as positive and negative internal representation, but it's very cumbersome. I'd rather call it emotion. Pain. Pain is an aversive internal representation associated with tissue damage. I'll talk a lot about pain today. Nociception, I checked the Oxford English Dictionary, and it tells me that nociception is the perception of pain. So a bit awkward. There's been traditional separation going back many decades between pain, which supposedly we're supposed to use only if the brain is involved. So for example, limb withdrawal from heat can be a reflex, therefore we should call it nociception. Not a great example. I'm not happy about this separation. And I don't think it's justified because in all animals carefully studied, the brain is involved as indicated by aversive learning and fear conditioning, which I'll talk more later today. I mentioned fear, so I'll also define fear. It's basically an aversive internal representation caused by the threat of pain or harm. So the overall logic of my talk today is I want to give you a bit of a flavor of mechanisms which are important in order to understand the evolutionary biology of pain. And of course, proper understanding of mechanisms requires understanding evolutionary biology. The mechanism. Most animals, perhaps all, have nociceptors that are sensitive to actual or potential tissue damage due to heat, cold, mechanical force, or chemicals. The mechanisms underlying nociception are remarkably evolutionary conserved, and it shouldn't surprise the biologists in this crowd. You've heard at least one talk about C. elegans. I don't need to introduce this wonderful model system. Nociception has been well studied in C. elegans. We see here 4 of the nociceptors that it has along the body. There are 2 more that you cannot see from this side, and then it has a few more in its nose. Fruit fly larvae also have nociceptors. I'll talk about the class 4 neurons later today. And this is a nice picture of them. Much of my talk will be about fruit flies. In part, I wanted to stay away from other speakers that you've heard earlier today. I like bees, but I decided not to talk about bees today. C. elegans, another example. So just to make sure that you don't have too much redundancy, I will focus on fruit flies. Why fruit flies? Of course, they're so beautiful. Don't you want to hug them? At least one person wants to hug them. It's my student, but maybe not. So why fruit flies really? Do I love fruit flies? The truth is I don't, but they are amazingly good model system with unbelievable powerful research tools, and I'll talk a bit about these tools later today. They do have a simpler nervous system. As Lars Chitka said earlier today, it's not simple. The larvae have only 3,000 functional neurons, but it's already a lot actually, and it allows them to do quite sophisticated behavior. The adults have about 100,000 neurons. Definitely not simple, but simpler compared to other animals, including humans, of course. Now, the research on fruit flies is highly relevant for all animals. One remarkable example is that almost all the mental impairment genes in humans have fruit flies equivalents. This is a remarkable number, and again, it's not an academic information that is removed from reality. Fruit flies have been used highly successfully for applied research relevant to humans. There is a large company in San Diego, Dart Neuroscience, that has been developing drugs for treatment of neurocognitive disorders. And the brain behind this company is Tim Tully, a fruit fly researcher who discovered 20 years ago that what he studies in fruit flies is highly relevant for mental disorders in humans. So as I said, saying that fruit flies are relevant to humans is not just some academic marketing. It definitely translates into results. So fruit flies have been studied for a variety of issues relevant to all animals, including humans. And Daniel Tracy, about 20 years ago, started to study pain in fruit flies. What he did, and fruit flies are a wonderful system to do it, he generated many mutants and basically probed each one of them to see whether they care about a hot probe touching their body. Of course, we would not like it. And what we see here on the left side is the wild flies that are used in many labs, and they respond almost instantly to the hot probe. And through such quite painful experiments by Tracy, he could find the first pain mutant in fruit fly larvae that basically failed to respond to pain. Compared to the quick response here, we don't see them, or most of them don't respond within 10 seconds. Further work by Tracy and colleagues on the painless gene indicated that it encodes an ion channel from the TRPA family, and not surprisingly, at least for me, the same mechanism occurs in all other animals, from C. elegans to humans. Inspired by this pioneering research, other labs adopted fruit flies for research on pain. This is a remarkable single paper authored by 42 others, and you'll see why. First, they relied on the amazing library of fly lines to screen over 16,000 different lines. This is a remarkably effortful research project. It's basically kind of industrial. And based on the genetic analysis of these lines, they identified close to 600 genes that could be implicated in pain. The paper don't get into details, but I'm sure it was a long process in which they decided to focus on a single gene called straitjacket, or STJ in short. And basically, after you identify through genome-wide association studies candidate genes, you need to verify that indeed this gene is involved in pain caused by heat. And what they did was generate two distinct lines. It's always good to have more than one for obvious reason. Oops, sorry. And tested them. We see the control line, the wild type, and we see that the two lines in which STJ gene was inactivated indeed responded more weakly to pain. So this is the fruit fly part of the study, but of course Neal et al. Were actually interested in pain and its relevance to humans. So what they did next is seeing whether the homologous gene in mice is also involved in pain. Obviously, mice are more similar to us than fruit flies. And when the generated flies shown here in blue with inactivated STJ gene, oh, sorry, okay, here. They indeed found, we look at the latency to respond to painful object, we see that they are slower to respond under a variety of hot temperatures. So basically, they established that STJ gene is involved in pain in mice, and then they did something even more remarkable. They moved to humans. In humans, you cannot generate such mutants for obvious reasons, but they could look at natural allelic variation in the STJ gene, and what they found that, so there are basically two major alleles, C and A, and people that are homozygous for the C allele were less sensitive to pain compared to carriers of the other alleles. So this was nice. And then they did another very nice study where they basically looked at the allelic variation in 169 adults 1 year after back surgery. And they basically asked them how much back pain they have. What they found is that the CC carriers, as expected, reported much less back pain. So what we've seen is that starting with a genome-wide screen in fruit flies, with the goal of identifying relevant genes and mechanisms of pain in all animals, including humans, was successful. So far, I know that they basically found the STJ genes. It may be that they're working on others now. Then they immediately moved to mammals and humans, and indeed found out that the very same gene is involved in pain and highly relevant back pain in humans. So this may be the most important slide I showed today. I basically showed you a remarkable example how inclusion is much more successful than exclusion. If we focus on the remarkable similarity among animals, it helps us understand all animals, including humans. There is just no logical reason, from simple pragmatic reasons, to be exclusive and say we are special. We cannot study other animals in order to understand us. They are different. They don't feel pain. Don't take it personally. Each of you is very special, but still, we need to appreciate the remarkable similarity, biological similarity among animals, and basically, inclusion has been successful, as I've already shown you. I want to move now and talk about the evolutionary biology of pain. So I already defined pain. It's innate negative internal representation. And basically, it elicits escape or withdrawal, rest, reduced motivation, sensitization, or avoidance learning. So you can have all these things. And the question is how pain is related to possible advantage or fitness in animals. There's been relatively little research about it, but I think it's a very important area of research. And I will go through a few basically hypotheses and summarize the available data about them. So number one is avoiding or minimizing tissue damage due to heat, cold, mechanical force, or chemicals. It seems intuitive, but we want to see data. This has been studied by Daniel Tracy's group at Duke University, and they studied it in the same model system, the fruit fly. Fruit flies, like many insects, have parasitoids. We see here a small wasp with a very long ovipositor, and this wasp attempts to lay an egg inside the larva, so her baby basically would develop inside the larva and emerge one day as an adult. Larva parasitoids can easily kill about half of the larvae in the field, and not surprisingly, larvae know how to respond to them. So what the parasitoid does, it penetrates the larval epidermis with a sharp ovipositor. It would feel to us like basically a sting or a needle puncturing our skin. And the larva has a very interesting response to it. We can see it here in the frames from a video. A bit difficult to follow here. The insects have 6 legs, but you see the ovipositor here, basically attempting to penetrate the larva. The larva responds very strongly, and it does something counterintuitive. It rolls towards the parasitoid. You can think about it, try to simulate it later. If it rolls away from the parasitoid, it would actually be easier to penetrate the larva. So it moves towards her. It causes the the ovipositor to curl around and might also basically make the wasp stumble, which you see here. It's out of focus, but because it happens quickly. And the larva here did it successfully. The ovipositor is outside, and the larva is basically crawling away now. Now, how is it related to nociception or pain? I already mentioned, I showed this picture earlier. We know that these nociceptors are used to feel the penetration of the larva. And the Huang et al. In Tracy's lab basically look to see whether larvae, we see here the response of control larvae with intact nociceptors, and we see that they respond very quickly by rolling and trying to escape from a noxious stimuli. On the other hand, if you inactivate the nociceptors, the larvae do not respond. So basically, we've linked nociception to indirectly to fitness. The larvae are more likely to escape death, basically, if they respond to the parasitoid penetrating the skin. So this was number one. Another possible adaptive significance of pain is reducing further risky exposure to danger and facilitating recovery through reduced motivation or rest. Remarkably, I know of no critical data relating to it, most likely because of very limited research effort to pain in animals other than humans. I must say that the optimal response is not obvious. We know from humans that sometimes keeping activity after injury is good. So it's definitely a matter of future research. Number three, another possible adaptive significance of pain, obviously all of them are not mutually exclusive, is reducing further risky exposure to danger via sensitization or avoidance learning. Just to make sure that we are all understanding each other, sensitization means increased response to stimuli following the presentation of a prominent stimulus. For example, we would likely startle more in response to some noise after watching a scary movie. Sensitization may be adaptive because there are typically spatial and temporal correlations in sources of tissue damage, and injured animals may be more susceptible to further injury because they're injured. I know of one example about sensitization in insects. It involves tobacco hornworms. The caterpillars are very sensitive to a poke on their body because, like larval fruit flies, they can be attacked by a variety of parasitoids. So if you poke a caterpillar, it responds very strongly by trying to hit the object that caused this poke, which in nature would be a parasitoid. The response is very quick and addressed to the very specific point of touch. So Walters et al. Usually working actually with aquatic animals, but here he did something with the caterpillars. He looked to see whether the caterpillars would be more likely to strike in response to touch, just relatively light touch, before and after sharp poking. We see here the number of strikes before, which makes sense. So basically, relatively light touch should not cause much response. But after poking, the caterpillars respond very strongly. The black and white don't matter here, it's just right and left side of the body. And then over time, it goes down. So the caterpillars definitely show sensitization. Here, I'm going to venture into non-insects, still invertebrates, but non-insects, because there are no data for insects, and this is a very elegant experiment done in squids. This is again from the Walters lab, and they were talking about nociceptive sensitization, and the idea is that you get sensitized after injury, and they provided the data. So what we see here, distance expressed in body length before and after an injury. So it's here before and then after. Uninjured don't change. The injured dramatically increase the distance from which they initiate flight. It makes sense because predators are fairly good at noticing an injured individual and basically cueing in towards this individual. And obviously the individual is more likely to be captured once a predator pursues it. So this makes a lot of sense. The clever experiment that Cook et al. Did involved 4 treatments. There was an uninjured treatment, uninjured plus anesthesia, which basically provided a very good control for the effect of anesthesia. Then there was injured, which basically involves pain, and injured plus anesthesia, which involves, I would say, no pain, less pain, definitely less pain. And the general prediction was that if you get injured but you don't feel any pain, you wouldn't be sensitized to the possibility of being attacked and eaten, and therefore you'll be more likely to be eaten under this case than this case. They used the real predator of squids, black sea bass, and I decided to skip a few slides because I decided this would be the simplest way to explain it. So basically, they put the squid with the predator for half hour and then looked how many survived. What we see on the left side is that the uninjured were much more likely to survive. So injury alone definitely has a cost. And we also see that anesthesia alone had very little effect, actually. So basically, the squids recover well from anesthesia. On the other hand, when the injury involves no pain, the squids do not sensitize, and therefore they suffer much higher predation. The reason is they basically let predators get too close to them, and then they're being eaten. So we basically show here the cost of sensitization to pain in a squid. And I use this example because, as I said, research effort to pain has been quite small. So I talked about sensitization, and I want to talk also about avoidance learning. And the idea is that pain can generate avoidance learning, which basically means, in this case, associating novel cues with the harming feature. Avoidance learning can be adaptive because one is likely to re-encounter the same harming feature, and learning about cues predicting harm can reduce further harm. Often in both human and non-human experiment, people use electric shock to generate pain. Obviously, it's a mild shock. It's used because it's highly aversive. We know it from humans. We see it in other species as well. It's easy to control and administer, which I like. It's not ecologically relevant, which I do not like. So this apparatus has been used now for about 30 years for testing for associative learning in adult fruit flies. And the way it works is we associate one smell, shown here in yellow, with electric shock or pain. We associate another smell with relatively good life. We switch back and forth between these. Then we move the flies to this elevator, move them down, and ask them where they prefer to be, the yellow or red. And the flies here do what they are supposed to do and prefer the red. So the flies can definitely show this behavior that I'll show you. This is a photograph from my own lab with my student Zach Dorisco doing such an experiment. And here's an example for result. For the results, we see on the left axis the proportion choosing the safe order, the one not associated with pain. The black shows the associative treatment. In order to test for associative learning, we also have to have very strict controls, which are presenting the flies in the apparatus only with smells without the shock, or in another treatment, only the shock without smells. And here we see, here we're interested for a reason I'm not going to talk about today. We compared females to males. No difference between the sexes here, but we see that the associative learning generated significant learning. The controls did not show learning, which we would expect. So basically, my lab, as well as most likely over 100 labs, have shown repeatedly over the past 40 years or so robust aversive learning to pain in fruit flies. Not only this, my lab and others, this is the first study, showed the same in fruit fly larvae. We're talking about the larvae with 3,000 neurons. The exact protocol is not important. It's very similar to the one I showed for the adults. Larvae are different, so they need slightly different protocol. And already in 1979, people showed that fruit fly larvae learn to avoid odors associated with pain. We see here the learning score of the wild type and it's much higher than random. And the important aspect of this research already in 79 was to identify mutants of learning in order to understand mechanisms of learning. But obviously, this is also relevant for us understanding pain. So I talked about a few possible adaptive significance aspects of pain. The summary of the available data, as one would expect given the limited research effort, is limited. There is some data that pain can increase fitness through escape or withdrawal. We don't know anything about rest or reduced motivation because it has not been studied as far as I know. Sensitization, there is one very nice study from the squids. Obviously, we need more, also in insects. And avoidance learning, the protocol has been used robustly for over 40 years, mostly for studying mechanisms of learning, but obviously we can extend it to understanding pain in a model system as well. So overall, there is limited data linking pain to fitness, but the reason is lack of research effort. Now, to truly understand evolutionary biology of pain, we need two things. We need to understand heritable variation in pain and heights associated in fitness. Associated with fitness. We know a bit about heritability of pain through studies on mice and humans here at McGill in the Mogil lab. And the heritability is around 0.5, which is quite good. In humans, you can use scales like the one you see here. In mice, there are good protocols also developed in the Mogil lab to infer through facial expression about pain. We also know, we don't know about heritability, but we do know that there is huge individual variation in pain relief in humans. And I'm talking about response to opioids, response to anti-inflammatory drugs that perhaps all of us have taken, and also placebo, which is a very interesting topic that I'm not going to talk about today. How about pain and fitness? In humans, there is some interesting research on a very rare phenomenon called congenital insensitivity to pain. And perhaps not surprisingly, the children, they are usually identified at relatively young age because they do lots of risky things, including self-mutilations that causes injuries. So we definitely know that humans that don't feel pain suffer. The good news is many of them, or the majority of them, manage to learn and live fine without feeling pain. So that's good to know. In non-humans, as far as I know, there is only a single study that I talked about, the squid study by Crook et al., that links pain and fitness. But as I said all along, the explanation is very limited research effort. Just for a variety of traditional reasons, it's not been studied. Obviously, first you need to appreciate the fact that you can study it in non-humans, and then hopefully people will study it. So to summarize the pain aspect of my talk, nociception is universal. Animals, including fruit fly larvae, learn to avoid pain. If you want to be conservative, replace pain with nociception. I basically said the definition for nociception is the feeling of pain, so I don't feel like doing it. Overall, we know little about the evolutionary biology of pain. So I didn't want to talk only about pain today, so I will finish with talking a bit about pleasure, mostly based on a very elegant recent study, which is in turn based on some very large body of literature in fruit flies. So pleasure is rewarding internal representation. And the focus of the research is ejaculation in fruit flies that has been well studied. And what we know that the neuropeptide corazonin induces ejaculation. The males have a group that is marked here in green. This is basically a green fluorescent protein that one can attach to these neurons, and the females don't have it. So this has been known now for a while. One amazingly powerful tool that is available for fruit fly research is optogenetic activation. So basically, one can attach microbial light-sensitive gene to a gene of interest, and then the gene would be activated only, for example, in this case, if you shine infrared light on it. And the use of infrared light is practical because flies cannot see it. In this experiment, males, so we see here the control where two sides were dark, and here the red, which would activate the corazoning neurons, and the dark. And what we see here is the positional preference. Basically, the question is, will the males that have activated corazoning neurons under the red light would prefer the red light? So we see these males here. When it is dark, they're not supposed to show preference, but when it is light, they're going to ejaculate. And the question is whether they are going to prefer the area where they ejaculate. We see here two controls where they don't show such results, and the basic message is that the males get some rewarding internal representation from being in the red light where the ejaculation is activated. The same very nice recent study by Zerkrispil et al. Also went further and asked whether males can show associative learning of an odor associated with ejaculation. They used the same established protocol I talked about before, and then you have the males under control condition and under the condition where the red light induces ejaculation, and we see that the males learn to prefer the odor associated with ejaculation. Now, you'll see in a moment why I talk about neuropeptide F. Neuropeptide F has been identified a while ago in fruit flies, and we know that it represents rewarding experience in fruit flies. Neuropeptide F, not surprisingly, is very similar to neuropeptide we have called neuropeptide Y, and some people, anybody familiar with neuropeptide Y? Okay, it's something that people learn when they take courses in motivation and emotion. Neuropeptide Y is associated, or most famous for its association with rewarding aspects of food. Now, we've known from before that flies prefer a zone with NPF activation. Again, it's optogenetic activation. And also learn to prefer odors associated with NPF activation. NPF, of course, stands for neuropeptide F. But the new study wanted to see whether corazonin is associated with increase in neuropeptide F in the brain. What we see here is two control lines, and the red indicates virgin males, and the blue indicates mated males, and we are looking at the expression of NPF in the male's brain as a function of their experience. What we see is an increase after the mate, but here when the corazoning neurons are blocked, we don't see such increase. what we see here is the effect of optogenetic activation of corazonin on neuropeptide F. So again, we're looking at the relative expression of NPF in the brain in two controls where there is no difference. And here we see that activation of the corazonin neurons we talked about before is involved in ejaculation, but it also causes a large increase in neuropeptide F in the brain. So the conclusion is that sex is rewarding in fruit flies through neuropeptide F. Ejaculation generates internal rewarding state, and males learn to prefer odors associated with both ejaculation and neuropeptide F activation. Overall, again, because of lack of research effort, we know very little about evolutionary biology of pleasure, but I hope I convince you that it's completely legitimate field of study. So to conclude my talk, we can and should study mechanism and evolutionary biology of emotion in simple model system because it can help us understand emotion in all animals, including humans. This is my last slide. I would like to thank you all for listening, and I'll be happy to take questions and comments.
I'd like to ask, all your examples are of animals with nervous systems. If we look at more primitive, go back to prokaryotes and single-cell eukaryotes, would you suspect that they experience pain in parallel ways, analogous ways?
Okay, so I'll think about bacteria. Bacteria would sense through receptors noxious stimuli and would turn away from them. So that's my answer. And I intentionally did not use a word you want me to use.
Well, Jenner showed all the way back in 1906 that eukaryote, single-cell eukaryote shows escape and avoidance responses to to what clearly are painful stimuli.
So basically, without mentioning his name, I know the research on bacteria quite well, and I basically said in my own vocabulary what they do. I'd rather not use the term pain. Now, between us, I talked freely about pain today, but I can tell you that, science is amazingly conservative because science is done by humans, and humans are remarkably conservative. If I had to publish anything, it's likely that I would not use the term pain, just because I need to compromise. But we can talk more about it, and I expect our thresholds to change over time, and I expect it to take longer than we like. So we don't disagree. It's just a matter of vocabulary and how far I want to go. Yeah.
Oh, hi. So I think based on my understanding, your definition of emotion is basically an internal representation that has valence in response to some stimuli. And then you go on to talk about pain, but there's also other negative emotions such as fear and anger, which based on your definition seems hard for me that internal representation alone can tell the difference between such categories. Do you think the context must be known in order for you to make distinctions between such categories?
I'm a bit puzzled by what you're saying. So you mentioned anger and fear.
Yeah. So, for example, for fear and pain, do you think they are different emotions? Or do you think that in animals such as fruit flies, they only have like primitive emotions compared to humans, so that it's not meaningful to distinguish between pain and fear? And
Yeah.
then you would just be happy to conclude that they're negative emotions, and that's all.
Yeah. So Lars Chitka mentioned today some data from bees. I used to work on bees as well. And I think that Lars and I would agree that bees show behavior absolutely consistent with fear. Now, because they show it, it means that we can find a correlate of this behavior in the brain, this behavior or this state in the brain. We have not found it yet. The same is for fruit flies. I basically talked, people vary in their vocabulary, but people definitely have used fear conditioning for the data I showed. So when you give flies electric shock and later ask them, do you like the smell associated with electric shock? The response is, so this is away from pain. So they have some internal representation that connects pain with a certain smell. And they basically avoid the smell, and I call it fear. So there is no question that there is some molecular way by which flies and bees represent a fear, and it's different from pain.
I guess I have a sort of related question. So you only talk about valence, right?
I'm sorry, I didn't.
You only talk about negative or positive valence when you talk about emotions. But in humans, like many people would also say other properties of emotions are important. For example, arousal and persistence, a lot of those things. Do you think that if I somehow get to measure those things as well in the fruit fly brains, that will make me tell those things apart? And also another one is that an internal representation of negative valence alone doesn't enable us to call it emotion, right? If I go out eat something that I don't like this food, I associate negative valence with it, but it doesn't necessarily mean that I attach a negative emotion with it. So where do you think we draw the line between? Yeah, I guess it's a difficult question.
To some degree, okay, so I definitely see where you kind of feel that my talk was lacking. I chose, based on logic and available data, to focus on the most accessible things, which are pain and pleasure. And even the pleasure, if the paper by Crispy et al. Was not published very recently, I'm not sure I could have talked about pleasure. So we're very limited by data, and I think it makes sense to focus on the simplest that would be less difficult to study. So the short answer is we don't know. I have no problem at all at accepting the likely fact that humans are more complex, more complicated. There is good biological reason to expect it. We know that when it comes to the brain, we have undergone at least two, since we diverged from fruit flies, basically the root of vertebrates has undergone through at least two whole gene duplications that resulted in many brain genes having more copies in humans compared to invertebrates, for example. So we have the potential to have much higher complexity. Another aspect is we live much longer. We have culture. So bees and flies are definitely super rich in behavior and cognition, but they live only about a week. It's very short. And obviously, it takes time to accumulate the amount of knowledge and complex cognition that we have. Um, so, um, so, so basically, I have two levels of answer to you. First, if we want to study emotion in simple model system, we better start with the things that are more accessible and then try to go further, and we might decide we're reaching a limit at some point. So for example, at the moment you do the screens in fruit flies and then switch to mice, you might find out that there are certain places where you cannot go further with fruit flies, but you can go with mice, and then you can do the same with mice. Very little of it has been done, actually. I have a paper from 2017 where I talk about it, where you basically can inactivate certain genes in mice and see how it affects their cognition. So basically, I'm saying pragmatically, we should start with the simple. And I'm not a philosopher. I'm very pragmatic, so I'm not going to say where the limit is. On the other hand, there are good biological reasons to expect humans to be more complex with more nuanced emotions. On the other hand, going back to the beginning of my talk, we know that we commit a crime by underestimating other individuals, including baby humans who do not talk. So we definitely have some flaw and bias.
Thank you.
Okay, I think the chairman should go, the moderator should go up front because they tend to have bilateral vision and speakers tend to look directly in front of them. There's two mics.
Oh, okay.
My question is not a terminological question, and it's not a philosophical question. It's a conceptual question. You, and you're not alone, put a lot of weight on the notion of an internal representation. Let me just take a second to say why most people do that. The simple idea that they have is, well, the two internal representations they're usually thinking of are shape and self. They say, when I'm talking about things having to do with sentience, I'm interested in whether the organism has an internal representation of self or internal representation of shape. We're not talking about that now. You are talking about emotion and pain in particular. Now, shapes are shapes. And the self is whatever the self is. So whatever's incoherent about this notion of internal representation, you inherit it with pain. What do you mean by internal representation of pain? What is the thing that you think is internally represented? And what do you mean by internal? I assume you mean by inside the head.
I can give the same answer talking about pleasure because it's better understood amazingly. I don't know why, but it is. So basically, I showed at the end of my talk that if you increase neuropeptide F in the brain, the flies like it. And we know that they like it because they prefer a location where neuropeptide F increases, and they learn to associate novel odors with increased activation of neuropeptide F. So in this case, I can say internal representation of pleasure in fruit flies is increase in neuropeptide F. Now, in mammals, it's more complicated, but coincidentally, there is a group at Concordia that studies sexual pleasure in mammals. They do lots of experiments on rats, and they would tell you exactly what are the internal representations of sexual reward in mammals are.
So do you mean nothing more by internal representation than an internal correlate?
I prefer to call it internal representation, or basically the molecular, or let's call it endocrinology and neurobiology that translate into a certain emotion.
I didn't understand that. How was that the answer to the question whether, do you mean anything else by internal representation than an internal correlate? And if you're talking about pain, you're saying the fruit fly is in a state of pain, and because I found an internal correlate.
So, yeah. So I understand you well. And I do not like the term correlate. My thinking about biology is that whatever we feel is driven by certain neurobiology and neurophysiology. So for me, it's not a correlate, especially if one can show very elegantly, experimentally, that, for example, neuropeptide F can generate basically sexual pleasure. So I don't call it correlate if you can show that it is necessary and sufficient to cause sexual pleasure. But to some degree, yeah, we can talk more about it. It could be some difference between a hardcore, highly pragmatic biologist, as I am, and somebody who may tend more into philosophy. And maybe we can move to another question by a philosopher.
Oh, well, he's first.
Oh,
Just
sorry.
don't forget to
Sorry.
come back.
Oh, yeah,
So, yeah.
Steve. Sorry, I forgot you.
That's all right. So, Reuven, this idea of emotion as an internal representation of valence. I mean, I think one thing that struck me, or that I wasn't clear about it in your definition, you can imagine a lot of things, even things without a nervous system, can approach or avoid stimuli, and that those stimuli could be described as having some valence to them. And I wonder if, in your idea of what an emotion is, there is built in the idea that that there's the capacity to update that representation. So when we think about emotion in humans, it's not just the approach or avoid, but it's all the connotations of the circumstance that we learn through experience. And even with fruit flies, the conditioning tasks and the ability to associate arbitrary stimuli with the valence. Is that ability to associate stuff with the valence essential to the experience in your view? Or is an emotion just the signal that tells you to approach or avoid it?
For me, emotion. Okay, so fruit flies alone have about 80 neuropeptides. I don't know how many mammals have. Do you know?
A lot. Yeah, I mean, and there are invertebrates that have many more than mammals. So yeah, I mean,
yeah.
the emphasis on chemistry versus electrical activity is maybe not essential.
But 80 is already a lot. The way I perceive emotion is it's basically an increase in certain simple chemical, neuropeptides are simple, that basically is a positive or negative, or there may be more nuances though. Okay, so for example, when the cat chases a rat, there is fear, and we can find the neural correlates of it, and probably neuropeptide that is involved as well. So that's my level of thinking about emotion. So basically, animals are hardwired and basically respond with negative or positive internal state. So there's no need for association.
I mean, it seems to me like the evolutionary function of emotion, and one that I think you would agree with, is to modulate your behavior in a way that's adaptive, right? But the whole notion of the modulation of the behavior assumes the capacity to learn aspects either of the motor output or of the sensory input. And without that, it just seems like what you really have is almost a reflex that wouldn't need to be called emotion. It seems like when you invoke emotion, that there's kind of a plasticity that's inherent in the idea that maybe doesn't come up immediately, but if you dig a little deeper, it has to be there.
So I see what you're saying now. I theoretically I can see how I absolutely divide between emotion and learning. In practicality, effectively all animals can learn. So you can overlay learning over emotion, and obviously you can gain more from learning. But if you poke my hand now with a needle, I would retreat, and I would say this is the adaptive significance of emotion. Without learning, every time you do it, if I retreat, I gain something.
If I don't want to take up all the time here, but
we have time, yeah.
But if the if you strike your patella and you have a reflex, it's adaptive, right? You're changing your muscles to accommodate that stretch. You're doing something there that's an adaptive modulation of your body in response to the environment, but there's really nothing that you would call affective about that, right? And I'm trying to distinguish between something that's a simple algorithm for an adaptive response and something that we intuitively mean when we say emotion. And I think maybe, and I never thought about this until your talk, that the learning aspect is an essential part of that. But anyway, we can disagree.
But you know what? And I'll just continue giving the pleasure example. Suppose you, okay, let's talk about fruit flies. It's easier. Suppose fruit flies move in the environment, and at some point, neuropeptide F increases in their head, and they say, "This is great. I'm going to stay here." So you don't need learning for it. So that's basically what I mean.
That sounds like learning to me. No, I mean, really, you've got the context of the world that they're in when they experience that, and that's driving them to be in that place.
We can talk more about it, but
Okay,
chemotaxis basically can generate what I said, and people would agree that chemotaxis is not learning. But to some degree, we agree that when you overlay learning on emotion, you can gain way more compared to something that is only theoretical because all animals do learn. But I see your point. Yeah. Yeah.
So that was a nice lead into my question because it seems to me that there's pretty good
I don't
evidence.
hear you well. Can you be louder?
Oh, talk more into the mic. So it seems to me that I thought that was a pretty good lead in because I think there's actually pretty good evidence in humans that certain types of associative learning can take place without people being aware of it, or at least without people being able to self-report any awareness of it. And so I guess my question is, in general, do you think, or if you're trying to make the sort of bigger claim that certain type of associative learning is indicative of conscious experience, it seems like you need to isolate a certain type of learning that can only occur with conscious awareness, and not just sort of any type of associative learning counting as a sort of example of flexible behavior. So I'm curious if you have any thoughts on what types of learning might fit that category.
I might disappoint you, but I have never talked about consciousness, and I'm not going to talk about consciousness. I don't understand consciousness, and again, I'm very pragmatic. I am very low at this point. You can aim high, but I have not done research on consciousness. Obviously, other people do it, but I just try to address way simpler questions for which we don't have answers. Sorry.
Okay, I mean, I think that's fair enough, but I do think most people's intuitive concept of pain is something that's an experience that we're aware of. So if you're saying, oh, well, all insects feel pain, you're sort of, for most people, giving the impression that there's some awareness there. At least that's the sort of way the conversation is used. So maybe there's a good reason for you to use nociception in scientific papers, if that's all you mean.
You know what? About a long time ago, more or less, during my PhD, when I talked about cognition in bees, people looked at me like I don't know. How come can you talk about cognition in bees? So now people have no problem with this. So we're changing slowly. With pain, if all animals have pain receptors, why do I need to use nociception for other animals and pain for humans? For cognition, for example, people say, oh, cognition is something special. Humans have cognition, other animals don't. So with pain, people made the same distinction, but it was completely artificial. There is no biological ground to separate between cognition in humans, cognition in other animals, or pain in human, pain in other animals. One day we might decide that there is reason, biological reason to do it, but what we know today, I don't attach consciousness to pain. Pain for me is a fundamental biological property. I cannot imagine animals living without the ability to perceive pain. And I don't like doing this artificial distinction between nociception and pain, especially because nociception means feeling of pain. So, yeah, maybe unsatisfying, but that's what I feel. Yeah. Yeah.
So some of this may be redundant because, but it'll be simpler.
But can you talk to the
For
microphone?
sure. Yeah. Some of this may be a little bit redundant because it seems to me like it sounds like the previous questions. Let me still give it a try. First of all, could you please clarify, just remind me what you mean by nociception? What is it to you? Is it the detection of pain? I thought I saw that before. Or is it the detection of like damage to the body?
So the hardcore physiological definition would be sense of injury or impending injury. So, for example, if you bring a hot probe close to your body, there's no injury, but there is a sense of imminent injury, and our nociceptors respond to it.
So I'm trying to understand what, as humans, I think we have been talking about psychological pain, so-called psychological pain. You know, when people are brokenhearted, etc. We even tried putting them in MRI machines. Apparently, recently brokenhearted people. Apparently, there was a study doing that, and it seemed like the pattern was the same, or at least it was very similar. But in order for that to make any sense to me, it needs to be felt. I mean, it's not about detecting that you're brokenhearted. You feel that it hurts, even though there's no damage to your body or even impending damage to your body.
Mm-hmm.
And so this leads to my question. It seems to me that nociception is not necessarily equivalent to the subjective feeling of pain that we have. And what's your opinion on this? And how do you think we could tell both apart? Or do you think it's useless to?
So I see exactly what you're talking about, basically emotional pain and its association with physical pain. And to some degree, Steve was here. I don't know whether he's still here. Oh, yeah, he's there. It came up with Steve yesterday when somebody, I think, asked him. Oh, okay, sorry. I'm sorry, what? Yeah, completely right. Somebody asked Steve about loss of partner in voles, in the monogamous voles, of course. And I think there's some evidence that it causes depression-like symptoms, which you can call emotional pain. Now, I have no problem at all with it because you already mentioned MRI shows certain neuronal activity. Personally, I like to know more about the chemical changes that MRI doesn't capture. So I have no problem with it, meaning I can imagine other animals having basically internal representation of loss. So for example, if a honeybee returns home to a hive and the hive is not there, I would expect to see some correlates in the brain of not finding home. So I have no problem with it at all.
All right. Well, maybe one last question.
Yeah.
As we know, the whole point of this whole summer school is to know if it's okay to hurt animals and if they actually feel it, or rather than detect it. So I think the general consensus is that most likely insects don't feel pain. You're saying that they might, but you're defining it differently than what we're looking for. So could you at least conjecture on how we could possibly reconcile that, you being the expert on insects?
Yeah. So to some degree, it's a matter of approach. So what I'm saying is inclusion is better than exclusion. And I think that by studying simple model systems like fruit flies, we can learn much more about pain. You want me to provide answers that, based on my pragmatic approach, checking the data, I cannot yet answer. And maybe being younger, you want to be, or you expect quick answers, but we need to appreciate that things are complicated and it's hard to understand other animals, so we need to be patient. So I'm not in a rush to provide an answer. And for example, consciousness, let's leave it to the future once we know more. Of course, anybody can study consciousness, and people have done well. Personally, it's too far from biology at this point, so I'll leave it to philosophers. Yes?
I enjoyed the talk. So the questions actually follow up a bit from the last two questions. The first one is about cognition. So you were saying that we used to be unhappy to talk about B cognition, and now we're happy to talk about B cognition. But isn't that partly because we've changed the definition of cognition to this quite broad definition? And it used to be that cognition in the 70s meant internal mental representations of things like cognitive maps, those kind of things. And now it's basically anything that goes on in the brain is cognition. So is there actually, what do you think about that?
Yeah, it's a good question. So again, my pragmatic approach was that inclusion is better than exclusion. And I don't like to have restrictive definitions that imply that we are special without foundational biology to use such restrictive definitions. So I like to start with inclusion, and then, as I said, the biological data definitely indicates that we're way more complicated, especially when it comes to the brain. We're yet to find specific characteristics that are unique to humans. One of them is obvious, our long-term experience and culture, but otherwise we have not found anything, and I think that Lars Chitka would agree with me about it. Okay. And you have another question?
Yeah, the other, so, well, one quick clarification first. So when you were talking about the adaptive evidence for the adaptive nature of pain, that was just the invertebrate literature you were reviewing, or you were reviewing everything there was?
I'm sorry, did you ask whether it's insect literature only?
Well, you were going through the evidence that pain was adaptive.
Yeah.
Was that just adaptive? Was that evidence just for invertebrates? Mm-hmm. Is there vertebrate evidence, or I just wasn't sure what you were reviewing. Were you trying to review all the literature or just the invertebrate literature?
Yeah. My focus definitely was insects. I did not kind of search my mind and literature for vertebrates. Yeah.
Okay. So, and then my question was a little bit similar to your question is, so what do we do if there is this adaptive variation and in how animals perceive pain? That seems to make things complicated in terms of applying that to animal welfare, partly because you might expect species to differ in how they perceive pain, and also you might see unintended consequences of treating pain, as you saw in the squid example. You treat pain, and actually the squid, their short-term welfare is good, but their long-term welfare is bad. So, what are the implications of that adaptive perspective for animal welfare?
Mm-hmm. Oh, well, that's an excellent question. And we can think about vertebrates as well. So, yeah, I talked about within species variation, but there is definitely also between species variation. To give you a very nice example, we do know that the expression of pain varies between species in an adaptive manner. So, for example, when you apply painful protocols to sheep that don't show any sign of pain, because for certain animals, expressing pain would attract predators. So in social animals, there is some benefit from expressing pain because you get social support, but in some other animals, there may be social as well, but in some animals, there's been very strong selection against expressing pain. So it didn't answer exactly to your question because it's basically a wonderful research question for research, but I don't think the research has been done. But you're completely right. I would expect variation between species. We know that there is enormous heritable variation in pain, meaning that there must be some trade-offs associated with pain and also pain relief, and we know nothing about it. Good, thank you. Good question.
Well, Mirsky's definition of pain, which comes from human pain, actually has an area that people have stopped talking about, okay? Because he said it's the expression of actual or potential tissue damage, or interpreted in terms of such damage. There's a genuine cognitive capacity in humans which must be addressed if we're going to think about pain. Think about placebo effect. Think about phantom pain. And I have the suspicion that when we're talking about, for instance, the pain of emotional loss of a partner, that it's this interpretation that is being brought out by human cognition that is laid on the actual experience, and that if we wish to understand pain in animals, we're going to have to figure out what that extra component is.
Is it a comment or a question?
Well, I suppose you can comment on the comment.
Okay, so yeah, I don't have anything to add about it, but yeah, maybe we can talk about it afterwards. So yeah, I don't have any immediate thing to say about it. Yes, Lars.
All right. So I know that studying consciousness is difficult, but I think you ultimately will have to take the plunge, because I think that is what makes the study of pain as a subjective experience interesting in contrast to just reflex loops. And well, so to give you a few examples, there's one example I already mentioned some days ago, I think before you arrived. So just a few days ago, I walked into someone's house and everyone sort of looked at my face horrified because I had this big splatter of blood on my front. So I smacked a horsefly just a few seconds earlier. I hadn't been aware that there was a horsefly. I had no recollection of having hit myself in the face. There was a perfectly adaptive response that got rid of the stimulus, so to speak. But it didn't enter my consciousness or indeed my memory. But there was clearly, I registered it at some level. The flip side of that is, and I think that's an example by Dan Dennett, where he reports an example of a carpenter who'd stepped on a nail in such an unfortunate way that it actually penetrated through his shoe and came out at the top. And the guy was in such distress and pain that they actually had to sedate him at the hospital in order to remove the shoe. When they did, they discovered that the nail had gone between the toes. There was no tissue damage whatsoever. So that's the fascinating interrelation between pain and consciousness. I only felt anything on my front after people looked at my face and said, "What's that?" Then I suddenly felt, "Oh, there's some." But so they're not the same thing. And I agree it's difficult to study in things that can't report on their experiences. But I think it's a necessary angle to understanding pain as a response, as opposed to just reflex loops, things that happen in bacteria, for example.
If you want to study consciousness, good luck. As I said, I'm pragmatic. I think that we have to understand more basic things before. I'm not saying we shouldn't and cannot study consciousness. I'm just saying let's start with some building blocks before going to perhaps one of the most complicated issues in biology. So that's the only thing I'm saying. Personally, I prefer to stay away from it at this point. I'm not saying that, obviously we know it's real, and obviously we know that it is a topic that should be subjected to critical biological research. I'm just saying, maybe not now. On the other hand, as I said, if your team is ready for it, go for it.
All right.
I'll support it, yeah.
I think that's an excellent way on which to end the session. Thank you very much.
Thank you.